Dr. Nhien from the Oxford University Clinical Research Unit in Ho Chi Minh City. As I mentioned earlier on, Dr. Nguyen is our precious collaborator in Ho Chi Minh City. She leads the molecular malaria lab at OCRU, and she has spearheaded the implementation of GenRe-Mekong laboratory analysis within a country. So just to give you a little background on this, until this year, really, our samples have all been processed at the Wellcome Sanger Institute in Kingston in the UK. Now, this is primarily because the process is technically quite challenging and requires quite a lot of sort of advanced equipment and so on. And the protocol is far from simple. And Dr. Nhien has worked with the Wellcome Sanger Institute in order to transfer this protocol to an endemic country. This is the sort of first successful implementation, and it is being used as a template for implementing in different countries, not only in the GMS, but also in Africa, in countries like Ghana, the Gambia, and so on. So with that, I would like Dr. Nhien to tell us about her experiences in doing so. Thank you very much.
Hello everyone, my name is Nhien. I'm from Oxford University Clinical Research Unit based in Vietnam, and many thanks Oliver for the introduction. And many thanks for the organizer for inviting me for this topic on this important scientific forum about genetic surveillance and one of the top trends in research recently. And my talk today is about building and implementing in-country laboratory for support genetic surveillance of malaria in Vietnam. And before coming to the main part of the talk, I would like to introduce you. As you see in the picture, it is the Hospital for Tropical Disease in Ho Chi Minh City. And this hospital is specific for tropical disease in Ho Chi Minh City with more than 500 beds and maybe one of the biggest hospital for tropical disease in the world. And why I introduced this hospital? Because our Oxford University Clinical Research Unit is here, 30 years old, and we are part of the hospital, and we are located in the laboratory department building with more than 300 staff working in many infectious diseases, including malaria, dengue, TB, CNS infection, enteric, emerging infectious virus, zoonosis. And our aim is to have positive impact on global health, especially in prevention, diagnosis, and treatment of disease in partnership with the Hospital for Tropical Disease, with local hospital, local research institution. And our main collaborators for malaria research in Vietnam is MP Nguyen. And you see in the next slide, they are laboratory and specific for molecular research diagnostic. And we have been working for many types of infectious pathogens. Our team are very experienced in whole genome sequencing of bacteria, viruses, and working with the GenRe-Mekong project, we have an opportunity to do the next generation sequencing with malaria parasite. And some of the key equipment have been funded by the GenRe-Mekong project to make our work more efficient. So it is the introduction to let you know more about our laboratory, and it leads to why our team is considered to be one of the team to implement the procedure locally. And as you have heard in Dr. Quang's previous talk, that dealing with anti-malaria drug resistance is one of the big challenges of Vietnam and neighboring countries. And there were the big changes in the way Vietnam monitoring the drug resistance in 2017 with conduction of the genetic surveillance in malaria provinces. And the big change is we conduct sentinel sign monitoring combined with the clinical trial, the TPS, and now change to large-scale surveillance. And from using individual markers to high-throughput genotyping. In collaboration with the National Malaria Control Program, especially in PPM, we conduct the surveillance in 12 malaria provinces, mostly located in central and highland of Vietnam. And the number of sites increasing by year. We start in 2017 with 30 sites, and currently it's nearly 90 sites in the region. And like Professor Oliver has introduced in the first introduction, the procedure, the sample collection is done simply and coordinated by the National Malaria Control Programs. The sample was collected from the health center and hospital. Then it was transferred to our group and sent for genotype analyze and generate the report. The report was then communicated with the National Malaria Control Program, hopefully to support the control and the elimination policy. But to dealing with the drug resistance, it is important to have the data timely and comprehensive. That's why it comes to the decision to conduct the in-country sequencing to support the surveillance. And in general, over the 5 years, we have collected sample in 12 provinces, and it is the overall status of the sample collection, as well as the proportion between the species, falciparum and vivax. And those data also was report to the National Malaria Control Program together with the genotyping and the drug-resistant genotype. So far, more than 4,000 samples was collected in Vietnam over the last 5 years and arrived our lab in Ho Chi Minh City. Back to the decision to perform in-country sequencing to support the surveillance. We have preparing for the 3 stages. First is training, then we test the procedure and comes to the operational procedure. We plan preparing and start the training in March 2020. And it takes a lot of effort of single team and our team here to check, to prepare equipment, consumable, reagent, primers, and review the SOP as well as the staff eligible for the training. And you may find the key information of the procedure published in the study resource of this genetic surveillance in GMS and South Asia to support malaria control and elimination, published last year by Chris and Oliver. In the study resource, many detailed information related to the procedure has been published. This is one of some of the pictures of the training. It is very strange that because during that March of 2020, it was a warning everywhere about the COVID pandemic in the UK and as well in the Asia and other countries. That's why we have to conduct the training straight away. All of the technical was communicate online with Dr. Shavanti and Mozam support. Then the lab work has been done by the local team. And on training, later by Oliver and Chris was done online. After that, we have the offline discussion with Shavante and Mozam to prepare to test the procedure in update. And one of the key highlights of the procedures, we prefer to have a high throughput, lower costing, and comprehensive genotype using the illuminamides. And the currently product target about more than 140 loci in the parasite genome, and we can process in one run 4 plates of sample, equivalent to 384 samples, and the sequence was done simultaneously. Back to the procedure, testing the procedure in Vietnam. We start the testing procedure after the training, and we completed in July 2021. It means it takes us more than 1 year to complete it. There's many works in the testing procedure, including testing equipment, testing with 1 plate of sample, calculation and balance the primer, update the SOP, QC, and discussion for troubleshooting with within the team and also with the supporting from the team. And finally, after several runs, we have the good result, and it is also was independent verification by Malaria and the team, especially on species detection and the genetic barcode. And when the procedure has done, we moved to the operational procedure from October 2021, we scale up to 384 samples per run with update SOP, more improvement on QC, advised by the team, and also we identify and we resolve some issue of equipment contamination and have discussion with the team and also Oliver team from Malaria. And after that, we are confidentially to perform the procedure in Vietnam. And previously, before we implement the sequencing procedure in Vietnam, we just received the sample registration and processing for the copy number variation in April. After that, all of the sample was sent to sender to genotype. And now, after we got trained and can run the procedure locally, the sample processing is also done in country app. And in coming soon, the cloud-based tool to help to analyze the data and also mapping to have a report and to help us to timely and quickly generate the data to report to national memory. To say about the implement the procedure in Vietnam. First, to make you more understand about the procedure. So far, we received samples from many sites in Vietnam, from Laos and Cambodia. All the samples were sent to Ho Chi Minh City, and these are the samples we received so far. And all the samples arrived approved, carefully packed. We check the barcode comparing between the document and the sample. The sample also has been done QC check and registered into a system called ROMA, sample management. And the PI from other study and country can track their sample from that sample management website. And it is a very complicated procedure. Before sequencing, we have to prepare the sample with DNA extraction, selective conjugate application, and then move to library preparation by several steps like amplification, tagging and adapter, purification, quantification, polling, and continuous sequencing. The raw data now has been uploaded And with support from Sangati was performed the genotyping and data translation. And from the releasing data, you can see, like Dr. Oliver said previously, there's the genotype related to drug resistance. And from the genotype, we can integrate and prediction to the drug resistance related to artemisinin, piperacin, laurin, butymetamine, and sulfadoxine, and as well nicotine. Also, the 101 SNP barcode generated from the genotyping can help to track the parasite population. And, the data also show the multiple infection of the sample and as well as the species of the parasite. Some of the results has been presented by Professor Oliver and Dr. Thuy previously, but I would like to highlight at this stage. Once we have the data, it moves to mapping and reporting to the National Malaria Control Group. And I want to highlight some use cases of the genetic surveillance. Once we have the data, we can understand the frequency of drug resistance that relates to the prediction of drug resistance in the region. As you observe in the example of what we have seen in 2 malaria seasons in Yala, that the genetic surveillance have to identify the hotspot as well as the dynamic of the resistance spread. Also, with some new drug is considering to use, like Dr. Quan said, in his amphotericin, nicotine can be considered as the last beacon. We can help to monitor the resistance to new drugs to see that the possibility of using this drug in the future. One of the other case is the genetic surveillance help us to have the warning of drug resistance in Dakhla in 2017 that lead to the TES was conducted in that province later. I thought that you remember in previous slide of Dr. Quan about the result of TES in many malaria endemic provinces that all the result has been done until 2016, and since 2017 with the warning messages from the genetic surveillance result from Dakhla, and the National Malaria Control Program decided to continue the TES in Dakhla and other related provinces. And you has observed in previous result of Dr. Quan about the result in 2019. And like the experience from Dakhla, we continue to monitor the resistance to the current treatment in new locations where treatment has not been changed. As you may know that malaria control program in Vietnam has decided to change to Paramax in several provinces with high prevalence of drug resistance. And there are some provinces has still UDP for malaria treatment, and we continue to monitor the drug resistance with the DP in those provinces. And you may see in this result, we also highlight the high DP resistance in some provinces near the coast like Thuy and Bintan in 2020. In summary, I would like to summarize some achievement we have got when implementing the procedure. However, I would like to say it with the challenges we face in each of the topics. First of all, genetic surveillance has been a useful tool to monitor the drug resistance, as you see in several use cases I just presented. However, the use cases is varied between country and region because it depends on the level of drug resistance in each area, in each provinces, and also state of malaria elimination within the country. And also, the implementing of in-country laboratory with timely and comprehensive support evidence for strategic decision making in malaria control and elimination activity for falciparum. However, it is a complicated genotype procedure, and it may require some minor optimization to adapt to local capacity. Also, it requires the supporting for troubleshooting for the team who have never done it before. And we also need the communication to share experience between local laboratories when implement the procedure. And additional markers, new drugs may be used for falciparum like combination with lumefantrine and modiatin may be added to the panel once the marker covers represented. Another point, currently the data analysis and pipeline is currently supported by single team, and we expect the timeline for the Laos-based tool readiness and update pipeline to release the result faster than that. And also because the data was primarily used to report and communicate to the National Malaria Control Program to support the decision making. So in Vietnam, it was the direct communication between our two in PPN and National Malaria Control Program. And in Laos and Cambodia, it is communicated to GenRe team in Malaria. So I think we need some more comments from the people of the National Malaria Control Program for improvement. And finally, one of the big challenges also, we would like to support the understanding of vivax epidemiology, drug-resistant elimination. And finally, I would like to thank for the sponsor and the collaborators, and most important, the National Malaria Control Programs of Vietnam and Laos and Cambodia also help to support us to implement the procedure in country, and especially the people. I will just highlight some of the people, but would like to thank many people for supporting us for the activity. Thank you very much.
Thank you very much, Dr. Nhien. That was a great overview of the many challenges that were faced in the creation of the GenRe-Mekong laboratory in country, which, on top of being a challenging activity in itself, was done in the middle of the pandemic, therefore without the ability of doing of the sort of training teams coming into the laboratory and all this type of stuff. So